Good day, grade 12 learners, and welcome to this video where we will be revising the first topic that you will be studying for your paper 2 of your NSC final examination. In this video, we will be revising DNA using section B questions from previous exam question papers. Please note that there are numerous revision videos that have been posted specifically for paper 1 as well. Please consult them and subscribe to this channel to ensure that you are going to be receiving all material that are posted so that you can revise for your NSC final examination. When you are going to be revising for the topic of DNA, the code of life, please ensure that you focus on the following important concepts in the topic. Firstly, using diagrams to identify the natural shape of each nucleic acid and arrangement of the nucleotides. Remember, there were different types of nucleic acids studied, namely DNA, mRNA, tRNA. Those were the different type of nucleic acids that we studied, especially when we look at the process of protein synthesis. You need to be able to describe DNA replication and know the importance of it. Describing the correct sequence, you need to be able to describe the process of DNA replication. The structure of a nucleotide, you need to be able to draw the structure of a nucleotide and identify the different parts, namely the phosphate group, the sugar, as well as the nitrogenous base. You need to be able to tabulate the differences between DNA and RNA, drawing the table with columns and rows and include correct headings at the correct places. Remember, distinguished when asked only nucleotides or the complete structure. If you are only asked to distinguish between the nucleotides, you only distinguish between the nucleotides. If you are asked to distinguish between the complete structure, you then focus on the complete structure. You will normally be asked to differentiate between DNA and RNA, either in terms of nucleotides or in terms of complete structure. The process of transcription, you need to be able to describe the process of transcription. You also need to be able to describe the entire process of translation. Memorize these processes and study it from your exam guidelines. Then you need to be able to answer one-word questions on transcription and translation, which we will look at today. The role of DNA and RNA in protein synthesis. Interpretation of diagrams of DNA profiling, very important. Refer to DNA profile bars or DNA bars and not just the black bars or just DNA. For example, DNA profile bars of suspect A compare with the DNA profile bars of the blood on the glass. Uses of DNA profiling. Uses for or against DNA profiling. So these are some of the key points which you need to focus on in this topic when you are studying for DNA, the code of life. Please ensure that you also know the brief history and discovery of the DNA molecule, the names of the scientists as well. That is also extremely important. If we look at the first question, 2.1, it says the diagram below shows parts of the process of protein synthesis. The process of protein synthesis. It does not tell us which phases we are looking at. We will need to look at the diagram to identify it. If we look at the diagram and we start on the right hand side, we can see that we have a single-stranded molecule here. That single-stranded molecule has become attached to a certain structure, an oval-shaped structure which we know to be the ribosome. The single-stranded molecule became attached to an oval-shaped structure known as the ribosome. Therefore, the single-stranded molecule at P is most likely going to be mRNA. We see that we have dotted lines here, and these dotted lines referring to base pairing. You have nitrogenous bases uracil, cytosine, and adenine, but they are coming from a major structure here. This is the nucleotide, but they are coming from a T-shaped structure, referring to as tRNA. This is the nucleotide of the tRNA. And here you are going to have all your amino acids of the tRNA. If we go to the first question, 2.1.1 says identify molecule R for 1 mark. So we go to molecule R. We already said that this is tRNA. We can see the structure of it. It is a T-shaped structure. Therefore, R is going to be referring to the amino acid. Then P, if we look at P in the diagram, P is showing us the single-stranded molecule here but became attached to the ribosome. Is going to now be known as the mRNA. The next question, 2.1.2 says write down the sequence of the DNA base triplet complementary to the codon Q. So if we have to go to the codon Q, here we have the codon Q. We already know it's on the mRNA molecule. Write down the sequence of the DNA base triplet complementary to codon Q. Now you are not given DNA anyway in this diagram, but you are given the tRNA anticodon. If you know the tRNA anticodon, it's then very easy to get the DNA base triplet because it's almost the same thing. The only difference is on a DNA sequence, you don't have uracil, you're going to have thymine. Therefore, the DNA base triplet will simply read TCA, and therefore that's the answer for 2.1.2. 2.1.3 state one significance of the nitrogenous base sequence on the mRNA molecule for 1 mark. One significance of the nitrogenous base sequence on the mRNA molecule. So we are looking at the nitrogenous base sequence on this mRNA molecule. What will this nitrogenous base sequence on the mRNA molecule do? It will give the sequence for the tRNA anticodon so that the tRNA can bring the correct amino acids that are required to form that particular protein. So it determines the sequence of amino acids that are going to form a protein. 2.1.4 explain the effect if there was a change in the sequence of the DNA base triplet in question 2.1.2 for 3 marks. A change in the sequence of the DNA base triplet in question 2.1.2. So 2.1.2, this TCA, if it changed to another sequence, what would possibly happen for 3 marks? Remember, if we change the DNA base triplet sequence, the mRNA codon also changes. And if the mRNA codon changes, then the tRNA anticodon changes as well. Therefore, the amino acid will possibly change, and if the amino acid changes, then it forms a different protein. Therefore, for 2.1.4, we will say there will be a change in the sequence of the bases on the mRNA, which may lead to a different tRNA, bringing different amino acids, resulting in the formation of a different protein. Alternatively, you could have said there will be a change in the sequence of bases on mRNA, which may lead to different tRNA, bringing the same amino acid, resulting in the formation of a different protein. Any 3 points for 2.1.4 would have given you 3 marks. 2.1.5 says name the organelle where proteins are synthesized in a plant cell for 1 mark. Notice how the question tries to confuse you by speaking about a plant cell. We are talking about the organelle where proteins are synthesized. Proteins are synthesized in any plant or animal cell at the ribosome. If the question had to focus on naming the organelle where DNA is located inside a plant cell, then you could possibly have said the chloroplast. But you cannot say the chloroplast here because the focus is on proteins are synthesized. And proteins are synthesized at the ribosome in any plant or animal cell. Therefore, the answer is the ribosome. The next question, 2.1, says the diagram below represents a stage during the synthesis of a certain protein. So you have different amino acids. You have your tRNAs carrying the amino acid as well as the anticodons. You have your mRNA strand attached to the ribosome. And you have numbers 1, number 2, as well as number 3. The first question, 2.1.1, says identify organelle 3 for 1 mark. This is organelle 3. What is organelle 3 referring to? The point at which the mRNA got attached to the ribosome. Therefore, the answer is the ribosome. 2.1.2, you're going to describe the role of molecule 1 during this stage of protein synthesis. You are describing the role of molecule 1. This is molecule 1. We are speaking about the tRNA molecule. You can see it's a T-shaped molecule. What does the tRNA molecule do for protein synthesis? It brings the required amino acid to the ribosome. So it brings the required amino acid according to the mRNA or the codon and takes it to the ribosome. The next question, 2.1.3, give the sequence of nitrogenous bases at number 2 for 1 mark. So we go to number 2. Number 2 is the anticodon on the tRNA molecule. Anticodon on the tRNA molecule needs to be complementary to the codon on the mRNA strand. So if the codon on mRNA is GGA, then the anticodon on tRNA needs to be CCU. On the DNA molecule that coded for histidine for 2 marks. So we go to histidine. Here it's the second amino acid. We want the DNA, the nitrogenous base sequence on the DNA molecule. Therefore, we can simply look at the tRNA anticodon. If it's GUA on the tRNA anticodon, it will be the same on the DNA base triplet. The only difference is uracil will be substituted with thymine. Therefore, the answer would be GTA. Therefore, the answer for 2.1.3b is GTA. 2.1.4, during the synthesis of the same protein, there was a change in the amino acid sequence because alanine was replaced by tryptophan. Explain the possible reason for this for 3 marks. So in this question, you can see they are obviously referring to a mutation because you had a different amino acid. Different amino acids are possibly going to form a different protein. But they're saying during the synthesis of the same protein, there was a change in the amino acid sequence because alanine was replaced by tryptophan. Explain the possible reason for this. So if we go to alanine, if we go to tryptophan, we can see that the tRNA anticodons are different. The tRNA anticodons are different. Why were they possibly different? Because the mRNA codons are also different, which means what? The DNA-based triplets are also different. So to answer 2.1.4, you first started by saying that a mutation occurred that changed the sequence of nitrogenous bases on the DNA strand from GGG to GAA. Then the mRNA codon changed from CCC to CUU, causing the tRNA to bring a different amino acid. And that is how you had alanine changing to tryptophan. So first we see the mutation occurred, which changed the sequence of nitrogenous bases on the DNA strand from GGG to GAA. How did we get that? Because this this tRNA anticodon will be the same on the DNA base triplet. This tRNA anticodon, GAA, will be the same on the DNA base triplet. Therefore, we say it changed from GGG to GAA. Therefore, the mRNA codon changed from CCC, we get it here, to CUU, causing the tRNA to bring a different amino acid. That is why alanine changed to tryptophan. 3 marks for 2.1.4. The next question, also forming part of 2.1, says 2.2, they are saying describe the process of transcription for 6 marks. Very important. This is something that will possibly be asked of you. Please make sure you answer it exactly as it is stated in your exam guidelines. Firstly, the DNA double helix unwinds. The DNA strands unzip. The weak hydrogen bonds are going to break to form 2 separate strands. But only one strand is going to be used as a template strand to form mRNA. Using free nucleotides from the nucleoplasm, mRNA is complementary to DNA. mRNA now has the coded message for protein synthesis. 6 points, but 2.2 would have given me 6 marks. Please make sure you are able to describe the process of translation as well as DNA replication as well. Those are possible questions which can also be asked for about 5 to 6 marks. The next question is taken from a 2.1 that says the diagram below shows part of protein synthesis. Now you see only part of protein synthesis. Why can we say that? We don't see the nucleus here. We only see the ribosome. We see mRNA on the ribosome. We see all our tRNA molecules coming along. What part of protein synthesis is it referring to? So based on the diagram given here, we can clearly see that this process, this part of protein synthesis, is specifically going to be that of translation. The first question here, 2.1.1, says identify firstly the part of protein synthesis that is represented in the diagram for 1 mark. Represented in this diagram, we just said the part of protein synthesis is going to be translation. Molecule S. So if we have a look at this entire diagram here, we can see molecule S. What is this molecule? It's going to be our tRNA molecule. Then the collective name for 3 nitrogenous bases indicated as R for 1 mark. So if we look at R, the collective name for the 3 nitrogenous bases on the mRNA strand is going to be referred to as the codon. The next question, 2.1.2, says write down the nitrogen bases at R from left to right with codes for glycine. So if we look in the diagram, we can see here glycine is the amino acid. Anticodon is CUU. We want the nitrogen bases at R. So R is on the mRNA strand. Therefore, R is referring to the codon. So we're going to change CUC to form on the mRNA strand. We're changing it to the codon. The opposite would be GAG. 2.1.3, we are describing the role of molecule S during the stage of protein synthesis shown in the diagram for 2 marks. The role of molecule S. Molecule S is the tRNA molecule. tRNA molecule needs to bring the correct amino acid to the ribosome for the process of translation. Therefore, molecule S brings the required or specific amino acid to the ribosome for 2.1.3. The next question, 2.1.4, says the DNA base sequence which coded for the mRNA TATCGGATG read from left to right. Explain the effect on the protein formed if the first T in the DNA bases was replaced by G and the C was replaced by A for 5 marks. So the DNA base sequence which coded for the mRNA is TATCGGATG. That's the DNA. Explain the effect on the protein formed if the first T base is replaced by G. This first T was replaced by G and the C was replaced by A. What would the new DNA base sequence now be? It would be GAT, AGG, ATG. That is what the new DNA base sequence would be. If you're explaining the effect that this will have on the protein, then it's only obvious that you need to explain the effect of this DNA sequence on the new mRNA codons. The new mRNA codons will now code for different amino acids. And different amino acids are going to possibly form different proteins. Therefore, the answer for 2.1.4, we first need to take this DNA base triplets and change it to the mRNA codon. So GAT will become CUA on the mRNA strand. AGG will become UCC. ATG remains the same because that was not changed. So the focus is basically only on the first 2 DNA base triplets because that was where the change occurred. So the mRNA codon changes to CUA, and the second mRNA codon changes to UCC. But remember, the initial mRNA codon would have been different. So you're going to state what the initial change was as well. So if we look at the answer for 2.1.4, the mRNA codon AUA will change to CUA. So AUA, the initial mRNA codon, changes to CUA because there we have CUA at the bottom. The codon GCC will change to UCC. So initially the second codon, which would have been GCC, would change to UCC. This will change anticodons UAU. Where do you get that? Initially here TAT would have given you UAU. And it changes it to GAU. Why? Because GAT now becomes GAU. And the anticodon CGG would have initially been CGG, changes to AGG. Because that's how the change that we had in the DNA base triplet. The amino acid leucine will be replaced by glutamine. So the initial anticodon that we had, UAU, would have given us leucine, but it changed to GAU, and GAU gave us glutamine. That's why we say the amino acid leucine was replaced by glutamine. Then, and proline will be replaced by serine. Why? Because on proline, we had CGG, and CGG changed to AGG. Therefore, proline is going to become serine, resulting in a different protein being formed. And that is how we get 5 marks for 2.1.4. The next question, 2.1, says the diagram below shows part of the process of protein synthesis. In this case, you can see you have process 1 and you have process 2. Process 1 is occurring inside the nucleus, and process 2 you can see is occurring at the ribosome. The first question, 2.1.1, says identify molecule B for 1 mark. Molecule B is this entire single-stranded molecule attaching itself to the ribosome, mRNA or messenger RNA. Secondly, process 2. What process is this referring to? The process of translation. Monomer D. We look at monomer D, the circle that's attached to the tRNA is going to be referred to as the amino acid. 2.1.2 give the anticodon nitrogenous base sequence of codon X for 1 mark. Codon X is UCA, therefore the anticodon is the opposite of that, AGU. 2.1.3 name and describe process 1 for 5 marks. Here we have process 1 occurring inside the nucleus. Process 1 is describing the process of transcription. So you name transcription, you need to have that there because you first need to name and then describe the process. So in describing the process, you're going to say the DNA double helix unwinds, the double-stranded DNA unzips or the weak hydrogen bonds break to form 2 separate strands, but only 1 strand is used as a template to form mRNA. Using free RNA nucleotides in the nucleoplasm, mRNA will be complementary to DNA, and mRNA now has the coded message for the process of protein synthesis. The next question, 2.1.4, says the table below shows the amino acids that correspond with different DNA codes. So you need to know your DNA codes in order to get your amino acids here. If you don't know the DNA codes, you can't get the amino acids. So write down the correct sequence of amino acids coded by codons X, Y, and Z respectively for 3 marks. So here you have X, Y, and Z. X says UCA, but we need the DNA code. So we change UCA. The opposite of U would be A, opposite of C would be G, and opposite of A would be T. Therefore, the first DNA base separate at X would be AGT. Then for AGG changes to TCC. And lastly, UAC changes to ATG. AGT, we go into the table and we see it gives us glycine. TCC, we go into the table, it gives us methionine. And ATG, according to the table, is going to give us arginine. Therefore, the answer is glycine, methionine, and arginine. 1 mark for each correct amino acid. The next question, 2.1, says below is a sketch of the DNA molecule of structure of DNA done by Francis Crick. So you have the structure of the DNA. 2.1.1, give the name of the scientist that worked with Francis Crick to discover the structure of DNA molecule. So in addition to Francis Crick, we had James Watson. 2.1.2, describe the structure of the DNA shown in the above sketch. Just for 1 mark, describe the structure. It will be the shape of a DNA double helix. The next question says that samples from an ill person showed 2 different kinds of nucleic acid in their cells, both the human DNA and the RNA of viral nucleic acids. The results of the percentage of nitrogen bases for the nucleic acids in the sample are shown in the table below. So you have samples 1 and 2, nitrogenous bases for A, for T, for G, for C, and for U. We notice that U is completely open here, and that's going to be in the first sample. But we also know that in a DNA strand, we don't have uracil. 2.1.3 says which sample, 1 or 2, contains the human DNA. We just came to that conclusion, there's no uracil here. Therefore, sample 1 would be DNA. 2.1.4, just explain one reason for your answer in 2.1.3 for 2 marks. So we just said sample 1 does not contain uracil. So you can either say in sample 1 the number of adenine is equal to the number of thymine. So you see here the adenine and thymine, it's both 21. That's one reason. Or you could say there's no uracil. So there's no uracil here in sample 1. DNA does not contain uracil. 2.1.5, you are drawing a simple label diagram to show the components that make up one nucleotide in a DNA. Very, very easy. The DNA nucleotide comprises of the phosphate group, the deoxyribose sugar, and the nitrogenous base. So that's all that you are drawing. Your phosphate group, your deoxyribose sugar, and your nitrogenous base, and you are labeling them correctly. 1 mark goes to the phosphate diagram, the portion here. 1 mark for the sugar diagram and the correct label. 1 mark for the nitrogenous base and the correct label. So 1 mark each gives you a total of 3 marks for this diagram. 2.1.6, you are tabulating one way in which the RNA nucleotide differs in structure from a DNA nucleotide. Only in terms of nucleotides, you draw the table, you get 1 mark for your table. You place DNA on one side, RNA on another side. DNA has thymine, RNA has uracil. DNA has deoxyribose sugar, RNA has ribose sugar. So any one difference would have given you 3 marks, 1 mark per statement, along with 1 mark for the table, gives you 3 marks. This brings us to the end of this video where we revised DNA, the Code of Life, using questions from previous exam question papers. So we completed the revision in the first section, DNA, the Code of Life. Please ensure that when you are studying for paper 2, you are studying the correct topics, that is DNA, the Code of Life, meiosis, genetics and inheritance, as well as evolution. The next video will focus on revising meiosis using questions from previous exam question papers. Thank you very much for taking your time to watch this video. I hope to have you all join me in the next video, which will be the revision of meiosis.